Today I'd like to talk about human genetic disease and its example where Bio 111 meets the real world. Up until now we've been talking about genetic disease in a very simple way, that we know a particular mutation causes a particular genetic disease. I want to talk today a bit about how we know that's true, and to do that actually we have to deal with some very complex issues. So suppose we have a person, and that person has a genetic disease, okay? And we want to know what mutation causes that genetic disease. So the first logical step might be to take a sample from that person, and you sample out some of their DNA, and you determine its sequence. And you find it contains lots of A's and G's and C's and T's. How do you know which one of those A's and G's and C's and T's is causing the disease? Which one's the mutation that's causing the problem? Well, certainly looking at the sequence of one person isn't going to be enough. You're going to have to do a whole lot of comparisons. And we'll talk about that in the next pieces. So certainly you're going to need to compare the DNA from more than one individual. Perhaps you'd like to compare a healthy individual with an individual with a disease. And that's a good place to start, but it has one fundamental problem, that individuals have many differences when you compare two different people. They look different, they have different ethnic backgrounds, all sorts of things like that. So you're going to find lots of differences between any two individual people, and it's going to be very difficult to figure out which is the one that actually causes the disease. So data from just two people is not going to be enough. You need to actually collect data from lots of different people and do a more complex set of comparisons. One particularly useful comparison is to collect DNA from many healthy people who do not have the disease and sequence their DNA, and look at many people that do have the disease and sequence their DNA, and look at what the people with the disease have in common that's not found in the healthy people. And so then you might find things that are unique to the diseased individuals and never found in the healthy people. And that's a particularly powerful technique to do, and is in fact oftentimes done. An alternative approach is to compare a single diseased person with many normal individuals. It's called measuring the allele frequency. And what you do is you take many people who are not diseased and you compare their DNA sequences, and you'll see many differences between them. And you can ask whether those particular differences are frequent, as you often find the altered base in many normal people, or they're infrequent. And the frequency analysis can give you some really good ideas about whether a particular mutation is likely to be causing trouble or not. And if you find that a particular mutation occurs at a high frequency in normal people, it's unlikely to cause disease, because those people are all normal. And if many of them have this mutation, that mutation can't be causing them trouble. So that's a way to rule out mutations. So if you find one of the changes and you suspect it might be causing the disease, but it happens to be really high frequency in normal people, that is, many normal people have it, it obviously can't be the one, or at least it's very unlikely to be the one that's actually causing the disease. So that gives you some more evidence. It's not perfect, but it gets you closer to an answer. Another very useful set of comparisons is to compare the sequences of humans and other vertebrates. The notion is vertebrates are other animals which are closely related to us. And so you can measure how conserved a particular sequence is. That is, there's a particular region of DNA where that DNA sequence is exactly the same in all different vertebrates. And the conclusion is then that it must be there. The reason it's conserved is there some sort of reason for that it's got to be functional, that a highly conserved base must be somehow important in the structure of the gene and the protein. And so mutations in highly conserved regions are more likely to cause a problem, because the idea is that base has stayed the same for whatever 400 million years of evolution or 50 million years of evolution. It's probably stayed that way for a reason, so messing with that might cause trouble. So it's kind of the reverse of frequency, that is, highly conserved things. If your mutation in your disease, or your suspect mutation, affects a highly conserved base, then it suggests that that's more likely to cause a problem, because you're messing with something that's been kept the same for millions of years. And so in class today, we're going to look at mutations in the context of whether those mutations are found at high or low frequency in people, and whether those are conserved or not conserved locations among other vertebrates. And those will allow us to try to build a case for whether a particular mutation is actually causing a disease or not.